Hello everybody, welcome back to our channel. Okay, if you have any idea about my video on biology looks, okay, please comment down. And also please, if you like our sharing, please like, share, and subscribe, okay? Today we talk about A1.2 nucleic acid, okay, SL and HL again. The HL is at the later part. First of all, we are still in A1, okay, unity and diversity in molecules, okay. First of all, we talk about genetic materials, okay. Everyone knows, okay. Genetic materials controls the characteristic of an organism. And DNA is the genetic materials of all living organisms on Earth, okay. And all instructions of organism to develop, survive, and reproduce are contained in DNA. So we want to have a closer look of DNA and the genetic materials. First, we look at the simple thing, the virus. Okay, virus is non-living, but it also has the genetic materials. A virus relies on its host for reproduction. Okay, and virus has a head of protein. Okay, and inside the head of protein, there's a nucleic acid. Mostly is a DNA, but sometimes okay, it has RNAs to store its genetic information. So it can be RNA, and majorly it is DNA. And then we want to study DNA, okay. And then the simplest part of DNA is the nucleotide, we call it. And then nucleotide, one nucleotide has three components, okay. Phosphate, pentose sugar, and the base. And to make it simpler, okay, we will have the symbol for it. The circle is for the phosphate. The pentagon is the pentose sugar. And the base is a rectangle. And all these three parts are connected by one single covalent bond. Later on, you will find a double covalent bond and triple covalent bond. Very funny. And a nucleotide has three parts, okay, phosphate, pentose sugars, and nitrogenous base, okay. And they are connected by a single covalent bonds. And a bond is very important because the sugar-phosphate bond between the pentose sugar and the phosphate is important because one pentose sugar is connected with two phosphate, and this bonding can continuous, and they form the backbone of DNA. So both DNA and RNA, you learn the difference later. They are polymers of nucleotides. Okay, then one such a polymer we call it a strand, single strand. Okay, and the sugar phosphate bonding is the backbones, okay, of DNA and RNA. They form the backbones of it. Okay, they two nucleides join together. This phosphate join with this the carbon of this pentose sugar here, and then they form the backbones, okay. And the pentose sugar and the phosphate of different nucleotides are bonded by the sugar phosphate bond, okay, to form a strand, okay. And the bases in DNA and DNA RNA has a difference in the base. Actually, we have 5 bases altogether, and the DNA has 4 of them, and RNA has 4 of them, okay, and 3 of them are common, A, C, G. Both of the DNA and RNA has the bases A, bases C, bases G, okay. So the difference is DNA has the bases T, and RNA replace bases T with bases U, okay. So altogether 5. So when you see bases U, it must be an RNA. When you saw bases T, it must be a DNA, okay. And in IBDP examination, in long question, okay, you need to have the full names of them. So try to memorize them. Really hard, but I'm sure you can do so. Okay, memorize each of them because in the structural question, sometimes they will require in the marking scheme for you to say name them, not just say A, C, G, T, U. In multiple choice, of course, you need to. Okay, just recognize them is okay. They have a different starting letter. Okay, A, C, G, T, and U. Okay, formation of RNA, or how do RNA form from DNA? Okay, DNA. So first of all, later on you learn a DNA double helix system. First of all, you need to single it out, okay, as a single RNA strand. And then this single RNA strand will become the template, okay. And along this template, okay, RNA will fit on it one by one. By who? By a RNA polymerase. So the RNA polymerase will start from one end and start to condense nucleotides, RNA nucleotides onto the DNA strand. Okay, so one by one. And then after that, okay, an RNA polymer is formed, and then they will separate out, and they are highly connected by the base pairing. So RNA is formed from DNA through RNA polymerase. Later on, we'll talk about a lot of this kind of steps, okay, and you get familiarized with it better. Don't panic. Okay. Oh, who is the first one who found out DNA? Okay, this is two groups of people contributors. First is an experimentalist. Okay, X-ray diffraction pattern of DNA. Okay, Rosalind Franklin do quick experiment. He stretched a DNA on paper clips, and then he shone the X-ray on it. Okay, and this strand of DNA diffracts the X-ray. Okay, and the diffracted X-ray on a sensitive paper, so we have the photo. Okay, and this photo is very important. We call it photo 51. Maybe he did a lot of successful thing. Okay, and then last, well done. Okay, she did. Okay, got the photo 51, and it is discovered in 1952. Not far away, not far away. So 70 years more. Okay. And then Quack and Watson theorists, okay, they look at this experimental results, okay, this X-ray pattern and a lot of different data. First, they look at the photo, and the other is that they look at some chemical studies of DNA, and then they try to make some models, okay, different kinds of scale models to fit the data, and they found that the double helical model fits all the data they have. So they think that double helical model is the most promising one, so they propose it in the same year as you see in 1952. Watson and Crick announced the double helical model of the structure of DNA. This story is fairly funny and controversial. You can go to the YouTube, not my channel, sorry, and you'll find out a lot of discussion about their relationship. These are two groups of physicists, their relationship. Okay, very funny. Okay, and then for the DNA structures, okay, DNA is a double helical structure, okay, so they have a two strand, okay, then two strand has an opposite direction. Can you see this one? The pentose up, pentose down, right? They are anti-parallel strands, okay? They join together. How do they join together? Linked by the pairing of the bases, okay? And remember, A is paired to G and G is paired to C. You see T, okay? So it's a DNA, RNA. When you see U, it is an RNA, A to U and G to C, okay? And then in the IBDP, you need to draw the two anti-parallel strand structure of DNA, okay? But our serious thing is about the bases, the length, and then the number of hydrogen bonds between them is a little bit our serious nowadays. And then a closer look of the thing, okay, difference between DNA and RNA. First of all is the pentose, the sugar are different, okay? For the DNA, it is a deoxyribose, okay? So less one oxygen, deoxy, okay? And for the RNA, it's the original one, R, okay? So hydroxide is here. So the name RNA, DNA from here, if it is deoxyribose DNA, if it's a ribose, low as a full structure, okay, OH, then this is ribose RNA. This is a difference, okay? Besides just the base difference, this is the pentose sugar difference, okay? Both have a helical shape, okay? But for the RNA, the other thing is RNA is only a single strand. So it's more flexible, okay? It can have different shape, okay? And the different kind of RNA too. It's mRNA, tRNA, and rRNA, okay? Very funny theory. You learn them details later. Okay, complementary base pairing. So the two strand has a complementary base pairing, okay? You can see they are highly connected. The C is connected to G and T is connected to A, okay? So if you have a one strand, definitely you know the anti-parallel parts, okay? It's parallel, okay? Exactly. So if you know this, the one sequence is like this, okay? You can do the pairing A pair with T, C pair with G, so that's we pay the A with T, and then you can see clearly the relationship, okay? And some part they'll tell you T and A are connected with two hydrogen bonds, and C and G are connected with three, but this is not that important, okay? In a Leo sequence, in Leo sequence is important. And then the possible DNA-based sequences. Remember, DNA molecules can have any length. Some are long and some are short. Really high variety. And DNA molecules can also have any base sequences. Okay, they like it. Although the two strands are highly connected, but for one strand it can have any base sequence, okay? Any. So DNA has an enormous capacity for storing data, okay? With a great economy. Later on, we'll know how to pack them up, okay? In other words, the diversity of possible DNA base sequences makes it the limitless capacity of DNA for storing information. So this information can be used to control the whole organization's growth, survival, or whatever, okay? And then we call it the conservation of genetic codes, okay? There's a concept called universal common ancestry. So it means that all the organisms on Earth come from one common ancestry, just one, okay? Because organisms use DNA to make amino acid, but all the organisms DNA use the same coding method, same coding, okay? Same codons, 3 codons for one amino acid, same codons, okay? Same coding. Seems that we conserve some genetic code, okay? And this kind of conservation of genetic code so that it is not accident. We come from the same universal common ancestry, although we haven't found it yet, okay? It is just a theory, okay? And then there's another theory about this thing, okay, genetic code, but in IBDP, you don't need to memorize anything. They'll give you the thing, they will describe it, and just need to interpret it and to understand it. Oh, SL is done, okay? Oh, now it's moving to the additional higher level, some more information about what we have learned. First of all, it's about the directionality of a DNA and RNA. You know, we said that there's forward and this is backward one, right? And then we call it from 3" to 5", okay? And then from 5" to 3", okay? It is the 2 strands difference, okay? So we call it leading strand is the 3" to 5" one, and the lagging strand is the from 5" to 3" one. This is the reverse one, okay? And the directionality is significant for replication, transcription, and translation. All are big things that you learn. Every word here has a big theory behind it, okay? And a complicated process. And then when you do the directionality of RNA, okay, when you do the directionality of RNA, when RNA is formed, remember it's formed from one end of the strand to the other, actually from the lagging strand, okay? Lagging strand 3" to 5", okay? For complementary, so the RNA will be formed 5" to 3", okay? But be careful, it is the lagging strands 5" to 3". So the thing is a little bit complicated, okay? So we use the lagging strand, it is anti-parallel to the leading strand, right? When we do this pairing, it is another anti-parallel thing, right? 5" to 3". So this is the anti-parallel and the anti-parallel ones, okay? So the RNA formed and the leading strand of the DNA are parallel. You got it? Very funny thing. Anti-parallel is parallel. So very funny. So remember, we start from the lagging strands 3" to 5". Okay? What we got, the RNA is 5" to 3". Okay? But it is the anti-parallel of the parallel. So it is a parallel to the leading strand of the DNA. And then the DNA has a helix system. It is a stability because they curl around each other, and the hydrogen bonds are very strong between them. Okay, so they can have a strong and rigid structure. Moreover, okay, they have some in the 3-dimensional structure. They have the same length. Okay, although AT and CG, remember some double bonds and some triple bonds, but they have the same length. Okay, so they're more stable. If they have different name, okay, some will be closer, some will be far away, okay, then it will be unstable. But actually, they have the same name, so it's a very, very stable 3-dimensional structures for the DNA. But DNA is long, right? So you need to curl them over to store them, okay? So we call it a nucleosome. They are curled around histones. This is a DNA double helix, remember? Then the DNA is very long, so they are curled around this 8 histone system. The 8 histone is a core, and after curling around it, okay, we have to bind them. It is bound by the H1 histone above it, okay? Okay, this is the system, and the whole thing we call it a nucleosome. It is a more effective way to store a very, very long DNA. And in IBDP, in the lab skill, you need to have the lab skill to use some molecular visualization software to study the protein and the DNA inside the nucleosome. There's a great picture. You can search it up from the internet, okay, to learn about it. But now you just need to learn that they are called around 8 histone, 4 a layer and 2 layers, okay? And this DNA are bound by a H1 histone, okay? Okay, funnier studies of in DNA is a very funny thing, okay? Some virus will attack a bacteria. You know, a virus cannot be produced by itself, right? It has to depends on the host, and this virus will inject its DNA, is the nucleic acid inside the bacteria, and take over the reproduction system or those, yeah, of the host, okay? And the host will reproduce the virus instead of the bacteria itself. So a lot of virus inside, okay, and then they burst out, okay, or diffuses out, okay, depends on, okay, and then a lot of virus is reproduced. Oh, thank you. That's the end of 1.2. I hope you enjoy it, okay? If you have any comment, please post it down there. I'll read it and I will reflect on it, okay? Remember, support us. See you. Bye.